In this figure, we're going to talk about protein structure and how it develops and how that is tied to the functions of these essential macromolecules. And those molecules can fall into both structural categories, things like cytoskeletal proteins, actin and myosin, or extracellular collagen, but also a functional category such as pumps and transport proteins or channels and enzymes. And so there's a whole variety of purposes served by proteins. Now, at the top here, if we start looking at how proteins are formed, and we have to remember that DNA encodes our proteins, and that is transcribed into mRNA and then translated into this sequence of amino acids. And what you end up with first is what's referred to as primary structure. And so I'm just going to circle one region of this protein at the top. And this simple sequence of amino acids, again, there are multiple different types of amino acids differentiated by their side groups. They're called the R group. But they have these different structures. And the order of those amino acids determines the primary structure of the protein. So you just end up with these beads essentially on a string connected by, in this case, covalent bonds. These are peptide bonds between the amino acids. And they actually allow swiveling around that bonding, which allows this strand of amino acids to fold over onto itself. And one of the first things that happens is we form what's referred to as secondary structure. And secondary structure is going to fall into two groups. First one here is a beta sheet. And the other one is known as an alpha helix, and that one's not shown here, which is essentially a spiral. But those secondary structures are formed by interactions between these amino acids that are occurring through those R groups. And so we end up with weak bonding, in many cases hydrogen bonding, between the side groups of those amino acids within the beta sheets and the alpha helices. Okay, well, we still don't have a functional protein. So what we need to have happen next is folding of this protein that has secondary structure. And that generates what's referred to as tertiary structure. And so in this case, one of the main driving forces of this continued folding is the hydrophobic residues, the water disliking residues. Again, based upon their R groups, they tend to not associate with water and want to associate with each other, with other hydrophobic residues, residue being the name of an amino acid within the protein sequence. And so we end up with these clustered, in this case, the hydrophobic residues are the solid circles here, clustered in the center of the protein away from the aqueous environment on the outside where the hydrophilic residues are, and they're happy to interact with the dipole within that water structure. Now, this will actually form, in many cases, our functional protein. This folding, it's very specific, ends up with one structure based upon the primary structure, the sequence of the amino acids. We end up with one folded version of a tertiary protein. And that will provide whatever function that particular protein is encoded for. It might be an active site. We could make this little pocket an active site here for interaction with some substrate or another protein. So we have a functional protein. Now, in many cases, though, we still don't have a functional protein at the tertiary stage, and we have to go to what's called a quaternary stage. And a quaternary stage forms what's called a multimeric protein. That's when we have multiple subunits, one here, one down here in this example. So 3 subunits are coming together, interacting through largely weak bonds to form one sort of massive structure, or larger structure at least, that has a function only when in that combined state. And you might ask, why do we need to combine these together? Why not just have it as one giant protein, right, that folds upon itself? And the answer probably lies in the fact that these individual subunits can interact with solutes, there could be solutes out here, that interact specifically with binding sites on these subunits. That then causes interaction between one subunit and another. And that allows for a more complex protein function. Hemoglobin, the oxygen-carrying pigment within blood, is a really good example of a protein with multiple subunits and that subunit interaction.